we start our discussion with for sequencing this week with next generation sequencing, and one way to refer to next generation sequencing is to say it is the high throughput sequencing. This is a class of modern technologies that are used for sequencing large stretches of DNA or RNA molecules. It's no question that the development of this kind of technology or technologies help to boost the experiments of biotechnology, biology, bioinformatics, and all different applications in different domains, giving the ability to scale up the level of experiments that can show us molecular biomarkers and molecular information, genetic information for many different species. Certainly lots of applications in health, in environment, and other fields. Now, unlike the traditional Sanger sequencing method, which we have discussed before, as you remember, Sanger sequencing methods focuses on sequencing a single DNA fragment at one time. Next generation sequencing as type of platforms can sequence millions to billions of DNA fragments simultaneously. That is what is meant by high throughput. Really go and scale up the way a single experiment is processed, therefore being able to process many other experiments. And so it is a massive parallel type of sequencing. And again, could be DNA sample or RNA samples, which are going to be through a process where they get fragmented. And then, again, we're going to speak about the different steps that go through in generation sequencing. Now, my first treasure hunt question today, which I'm going to ask quickly and discuss, but again, ask you to come up with a couple of more ideas. What is the difference between next generation sequencing and microarrays? Both are powerful technologies used in genomics, molecular biology, but certainly there are some differences. Okay, so what I want you to do is go and look up some 3 differences between these 2 technologies, whether you want to highlight the principle of how they work, or applications, or cost, or the type of sequences they can find. Share that with me, okay? Just to make sure that we're establishing understanding of a new concept, as well knowing what could be the differences between some. And anyway, just briefly, next generation sequencing is looking at DNA or RNA molecules. It can sequence millions to billions of fragments simultaneously. I mean, this is, again, the statement I'm going to repeat. It is for high throughput. When we look at the microRNA, which is based on hybridizing labeled DNA or RNA to the solid surfaces, it can handle thousands to millions. So we're talking about scalability of millions to billions, and here we're talking about thousands to millions. I mean, this is one of the differences, but I want you to look up a couple more and then share it with me as part of your answers. So what you see here is how the cost per raw megabase of DNA sequence, if we want to establish this as a reference to see throughout the years, specifically, this is, by the way, from National Institute of Health, from the NHGRI Institute, which we talked about when we highlighted the Human Genome Project. And again, this is, if we're going to look starting from roughly 2001, close to, if you remember what we're talking about, the Human Genome Project, right? So Human Genome Project was complete in 2003. And then how the expenses moved from there on. There is a drastic drop throughout the years in terms as we progress until 2021, and until this day there was a great progress to the level where sequencing one genome, a human genome, is going to be less than $1,000 compared to the first human genome, which costed something roughly between $1 billion to $3 billion, depending on the different reports. So $1 billion to $1,000, that is a drastic jump in a matter of roughly 20 years. And compared to the well-known Moore's law, which explains, the computational powers, how it doubles every year by adding numbers of transistors. It's the power of CPUs. So this Moore's law, it's this law to describe how our computers gaining power and processing. And so just a reference to show you that really what we had achieved throughout cost per megabase, raw megabase, or per sample or per organism has dropped dramatically, which means that nowadays it is accessible to very good extent. Now, still, if you're designing an experiment, you end up saying, well, I need 20 to 30 samples, right? I need to get my hands on 20, 30 samples from some species in the environment, some project I'm working with. Remember before we discussed this, you need to have some replicates, some biological replicates. So you need to go and have some samples to establish some statistical evidence that you are looking for to support your hypothesis or not. So you need some repeats, you need some groups. You may have some control groups, some different treatment groups. So you have a few samples from each. So you may end up roughly with 20, 30 samples per one project. And then again, it depends on the fund you have. So which is, if we are looking at 1,000, 1K to 2K per sample, it's going to be ending up of having something between 30K to 60K, 30 to 50K, $1,000 for one project, I would say, for one experiment that you're looking at. So again, it is drastic development. It's amazing. We have now accessibility to many of these technologies. We're going to discuss next generation sequencing in depth. I've referred earlier, I have a colleague working now on the 1 million sequence project in the States that's announced as part of Broad Institute and other institutes in the States. So if we keep going like this, then what we're looking at in the future, perhaps a couple of $100 per one experiment in the probably coming few years. Okay, I don't know. It could be like in 10 years from now, sequencing one sample is going to be $100 each person, and then we can scale up then the community and population experiments and move to a different level where we can examine diseases, genetics, and so on and so forth. So this is such an amazing technology that we need to look at and understand some principles about it. So what we see here is again from 2000 to 2014, 2015. In this figure, what you can see is there are two curves. One is giving us the cost per gigabase, okay? And again, you can have it for raw megabase, gigabase, and see how it's going to drop. And then let's look at another thing, okay? The output per week. And this is kind of telling us the throughput, how many experiments we can run, how many samples we can screen, and so on and so forth. So in the beginning, these are the names of different technologies, okay? And so in the beginning, it is the cost, like look at this technology, the cost is huge, okay? And the throughput is very low compared to where we are now, like HiSeq X10 technology system. It is the world's first technology to break the 1,000 genome barrier for human whole genome sequencing, okay? For human whole genome sequencing, okay? It is the first technology to break the $1,000 barrier for a human genome sequencing. So this technology, if you have it now, you can really, do screen a lot, okay, and for very low cost. And gigabase per week is huge, okay? Now, the technology itself might be costly and maybe even more expensive than before, but again, it depends on what technology you're looking for, different factors. So it doesn't mean that if we can screen one person that we can easily buy these machines. They are usually expensive. but it's a different factor that we look into. What we are happy with is certainly if there are genome centers or genome centers and national genome centers or laboratories or research funds or other types of funds, then we can go, and such machines are there and the right setup, we can run more experiments and then explore more. It is now not too difficult to ask to have more samples. Okay, let's have more samples, and it's becoming standard to have many biological replicates. So when it comes to Illumina sequencing, as any kind of sequencing, as a good example of next generation sequencing. So once we start talking about Illumina, okay, it is this very famous example from for next generation sequencing. There are other companies, other machines, and so on and so forth. We're looking to Illumina as one of the very known, recognized machines in different laboratories produced by Illumina. So Illumina sequencing, or this, I mean, this type of technology has some core ideas. So I'm going to introduce some ideas here, some terms, as we move from the complex DNA to, of course, always, what is the idea always? We have some surface, right? And there is some designed kind of, remember, we looked at these before when we looked, we used oligonucleotides probes. So there are some chips, some surface, solid surface attached to it, some designed nucleotides. Eventually, you want to see if those would bind to samples of DNA. There are going to be steps of course, fragmentation, cut it down, right, and polymerase chain reaction, amplification. So we discussed in the Human Genome Project some ideas, and we looked into, as part of the previous lecture on sequencing, and even before we looked at PCR, microarray. These concepts of fragmentation, amplification, binding are going to be always key concepts for these different technologies. Yet there will be some differences, and there will be some unique ideas related to specifically next generation sequencing that employs the methods like sequencing by synthesis, which is something we highlighted before. But today in this lecture, we're going to dive more into this. The human genome sequenced annually, now we're talking about HiSeq X10 from 2014. We're looking at roughly 18,000 genomes sequenced for humans every year. Compare that to barely one genome in 2005. So in a matter of 10 years, and nowadays, of course, this number is, again, we're talking about million, right? Different countries are on the track to do their own genome sequencing, genome centers. We're looking at one species, the human here. There are projects on different species and animals to look at the environment, and it depends on the type of projects that different countries are working on. Things sometimes related to food, for example. I was aware of research projects in China to enhance the quality of the flesh of fish, for example, which is going to relate to produce and exportation of these products to different countries. So this is an economy by the end of the day. So looking at the genomes could contribute to the economy. And so nowadays, this is really scalable as compared to 2013, 625 and hundreds. Now we're looking at thousands, and soon in the hundreds of thousands and more. So in Illumina sequencing, the idea of fluorescence is still there. Of course, they were used now to this idea. So what you see here, three steps, which we refer to as nucleotide addition, imaging, and cleavage. Okay, nucleotide addition, imaging, and cleavage are central to the sequencing by synthesis. So this is the general concept of sequencing by synthesis, and these steps are also part of Illumina sequencing. So when you look at the nucleotide addition, Illumina itself uses specifically designed nucleotides that are attached to a unique fluorescence dye. This is a unique fluorescence dye for adenine, for example, for guanine, another unique fluorescence dye. We're now used to this, right? We have different dyes. This is another fluorescence dye. So F for fluorescence dye, green for cytosine, and then red for thiamine. So each one of these four nucleotides are having its own specific fluorescence dye. Okay, and those nucleotides in Illumina sequences are also modified such that once they are incorporated into the growing DNA, like here, okay, once they are incorporated, okay, they temporarily block any further nucleotides. Okay, there is no other nucleotides that can bind here because we need to take a snapshot, which is an image. We need to take an image at this stage and see what colors we get, okay? And then this is going to define our current read of these different strands at this moment, okay? In imaging, we'll see some examples as well. There's what's called the flow cell. It is this, again, you can think of it as a chip. This flow cell is imaged through using a specialized camera or some light source or laser source to try to read the different lights. At the end, okay, after imaging, this fluorescence is detached and the blocking group are both cleaved off. So leaving behind just this added nucleotide, and then the process continues. So when it comes to one of the things that makes Illumina sequencing unique is the way it does the amplification. And here we have some interesting concepts. Okay, we're going to look at what is called bridge amplification. It's really nice, interesting. It is something very unique to Illumina sequencing. So let's follow up here with this, and then we're going to watch some animation. So to make it clear, it's not too difficult to understand. What we're looking at is that there is an amplification step to have sufficient number of identical DNA fragments. Okay, so this is the flow cell here. Okay, this is what we refer to as the flow cell. So it is where the amplification happens at the surface of the flow cell, which is coated with the oligonucleotides primers. Okay, so what you find here is two types of oligonucleotides primers, one in red and one in blue. You can see these colors here. Okay, so at this flow cell itself, there are different bluish and reddish primers, oligonucleotide primers, meaning short DNA sequences that are going to act as complementary to what is called adapter sequences. So what you find here is a template sequence coming from the DNA, and each DNA in the experiment, during the experiment of Illumina sequencing and part of the library preparation, DNA or RNA is broken into small fragments, the ones you see here in the middle. Okay, so imagine you will have many of those in the middle here. Okay, and each of these sequences, they will have attached to both ends other DNA sequences that are called adapters. They are short DNA sequences. So in this case, this is one adapter, and this is another adapter. This is one of the key concepts to understand when it comes to Illumina sequences. Okay, there are short DNA sequences, adapters for short. We did not use this term before. We used primers, templates, and so on and so forth. What we're looking at here is what's called adapters. Okay, adapters are attached to both ends of the fragment the DNA fragments. So we have more, and there will be more adapters, okay? So this is a nice way to draw the adapters that will have some added information here, okay? These adapters are crucial for the sequencing process and for the subsequent data analysis as well. So we're going to come at the end, we will look and see that even the final output we need to have some information about the adapter, okay? So what we were saying is that, again, the red part here, okay, is not the same red part here, okay? This could be the one confusion when you start looking at this part, but I'm going to speak about it. These are coming template binding, so these are going to come from the sample. We wash over the flow cell that we have here, which have primers. So what's going to happen is that there will be an adapter. The adapter is going to bind, okay, to the primers, okay, because these primers here that we have, these primers here are designed to be complementary to the adapter sequences. Those adapter sequences that we previously ligated to the fragmented DNA as part of the library preparation. So as a prior design, we have primers. So remember, these are the primers, and these are the adapters. And as you see, once they are washed here over the flow cell surface, they will bind to each other, okay? So this is the first step here. And then again, when the whole DNA library is loaded onto the flow cell, the single-stranded DNA fragment bind to the complementary primers on the flow surface side, okay? Now, bear with me for what's called the bridge formation, okay? Very interesting concept, bridge formation, bridge amplification. So after binding, the DNA fragments bends over to form a bridge by annealing to a second primer on the flow cell. This is where the bridge amplification comes from. So certain conditions of temperature, which we remember before we discussed, okay, we can refer, increasing the temperature is going to be meaning annealing. Now this is what you see. You see this piece here is going to bind, and then the primer here, okay, is going to also hybridize or link to the adapter, okay? So we will see this in action. When this happens, a certain polymerase reaction is going to happen, and then make a copy of these steps and then end up generating. So it's a very efficient way to end up generating many of these strands, detach them, and so in each, after several rounds of amplification, we get 100 to 200 million clonal clusters, okay? So this kind of clustering information is key part and step. There's going to be, after bridge formation, DNA polymerization, denaturation, and these are going to be detached. And the animation we'll see next is going to be very helpful to see the whole step. And once these steps are repeated for several times, after some many cycles of bridge amplifications, they're going to have millions to billions of dense clusters of amplified DNA fragments that are generated across the flow cell. Now, each cluster contains approximately 1,000 to 2,000 identical copies of a single DNA fragment. Each cluster, so there will be millions of clusters, and each cluster is going to have 1,000 to 2,000 identical copies of a single DNA fragment from the original library or the initial library. Now, one question that I want you to answer as part of the treasure hunt: why do we need this clustering step? Why do you think this clustering step would matter? Okay. In other words, what we mean by clustering is that we are grouping similar fragments or information together. Okay. And I just told you each cluster is going to contain 1,000 to 2,000 identical copies of a single DNA fragment. So we can rephrase this question and ask you, why do we need to have 1,000 to 2,000 identical copies of a single DNA fragment? So think about it and share with me your answer. So let's watch this very nice animation that's going to explain the process, and it's going to be very helpful to visualize everything we have.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, denatured.
So I'm going to repeat this here so that you get a hint, because the idea of the bridge formation at the beginning might seem, you know, where does it come from, right? Well, when you look into this, you see how this is going to be key to speed up the process, right? And this is what makes, again, this whole technology unique. It's the speed, the scalability. We can work now with millions to billions of copies. So maybe this, imagining this and linking it to the previous question, can help you see. So I'm going to repeat this small part to see how it links, may link to speeding up the technology.
Complementary strand forming a double-stranded bridge. Strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in base-based Sequence Hub.
Yeah, I guess this is very interesting animation that we have just watched. We looked into the idea of the different steps when we had the the DNA fragment with the 2 adapters hybridizing to the primers that are designed the flow cell through a process of annealing. This is going to bind to the next primer, and then a DNA polymerase reaction is going to happen, and so get the complementary, and then we end up with reverse strand and forward strands. Okay, this is going to be repeated until there are clonal copies, many copies and clustered copies of both forward and reverse strand. Very interesting. And then once these copies are there, and yeah, I guess it's important to ask this question. I assume we all are on the same track, so we know why do we need many copies, okay? So why do we need many copies? And this is another treasure hunt question, the third one for this part, and this is very important question. If you don't understand this question, make sure you look to the previous module, because we need many copies for different reasons. We have discussed that in depth before. So again, share with me a few ideas or thoughts that you think, why we are even doing this amplification at this stage. Remember, the Human Genome Project, we had some discussion about also different philosophies into how to do that. This amplification is very important. They end up with many copies. And then eventually, as you saw there, we're going to work with what's called alignment. Alignment is more of a computational process. It is a computational process that will require to have some reference genome. And when the time comes for my lecture on RNA-seq, we're going to see how we can process this kind of data using some tools. That is the alignment step. So sequence by synthesis as technology goes through different steps. And again, this is highlights here some of what we saw in that animation. You have 4 nucleotides with 4 fluorescents competing to come here, right? And then these are what we referred to previously as the DNTPs. We have four colors. Once we get the color excitation here, we can, through the wavelength, we can know which color is that, okay? And eventually we start getting the different reads detached. And so we have these different reads reported using this technology. Okay, now part of this is what's called reversible terminator based method. Okay, reversible terminator based method. Okay, which is a foundation and aspects of the Illumina sequencing. In traditional DNA sequence synthesis, once a nucleotide is added to a growing DNA strand, the next nucleotide can immediately be added. However, what's unique here in this reversible kind of terminating or reversible terminator technology is, for sequencing purposes, Illumina is going to have these specially designed nucleotides that are called reversible terminators. Okay, they are the nucleotides that are modified to have two things. Okay, two things: the fluorescence dye for detection. Okay, fluorescence dye for detection, and another thing is a terminator group. Okay, a terminator group, so it's going to block from other nucleotides to come, and this helps in enhancing the accuracy. Okay, doing the parallel sequence, because this method is going to allow for millions of DNA fragments to be sequenced simultaneously on a flow cell. So each sequencing cycle, that's what you call a different sequencing cycle. Every time, it's think about these lanes on the flow cell. Once there is one nucleotide added for each sequence, each sequencing cycle is going to read and try to image that through some mechanism. Okay, and results. Each sequence cycle is going to results in the addition of just one known nucleotide to each fragment. So when we speak about this step here, okay, we have added this nucleotide, and then once we move to add another nucleotide, we are talking about second cycle. Okay, now, sorry, second, or let's call it cycle 2, and then cycle 3. Okay, cycle 2, cycle 3, and we keep adding. Now, one may say, why we call it one cycle? Because it's not one strand. Remember, we have many of those, millions of these copies. So one addition, that's going to help to, and then control it, okay? Because if we don't handle this carefully, where we have many of these, millions of these copies, and we don't do it carefully, we may end up with, different noisy parts. So from different areas. This could be, if this could be longer, this is shorter. Think about it. This is long, very short. So the light is going to overlap on top of each other, causing some noise, which we are going to discuss as some limitations, because this kind of issues may happen. And again, think that I'm standing on, I don't know what's going to be the best way, but think that I'm stopping here, okay? And my kid is behind me, and he's also trying to shoot some light, okay, with his hand, and I'm trying to shoot some light, okay? And you are standing here, okay, and then trying to observe which light is coming to you, okay? You're not going to see the light of my kid, right? So if we don't add the one nucleotide at a time for all of these strands, make sure we are of the same, okay? But now imagine we have the same height, then you're probably going to end up seeing, probably, again, not always, but hopefully to a good extent, you'll be able to see, oh, this is green, this is red, and so on and so forth. So this is for animation for every cycle. You see one cycle is going to get, as this keeps going, you'll have different fluorescence types of colors coming out and through the imaging technology. So when it comes to Illumina sequencing, we have some steps that we are looking at here. There's the library preparation, okay? And again, there's fragmentation, ligation, where we end up having adapters in both ends. Then we go to the cluster amplification, and then that is part where we, based on that bridge amplification cycles, we end up having millions of these copies. So in terms of sequencing technologies and the different types, next generation sequencing is not one machine, it's not one company, it's not one entity, okay? There are many of these technologies, okay, out there. You can see here some names. For example, there's NextSeq, HiSeq, HiSeq X, NovaSeq, and so on and so forth. Each one could have different purpose. We made a highlight for HiSeq X series, because it's the first one, again, to break the 1,000 genome barrier. And hopefully we get, I'm not aware of anything to get the $100 barrier. I think a few years ago, we met somebody from one of the known companies, trying to just on top of my head. They were part of recent technologies, and I guess we're going to see $200 very soon, especially with minions, these very nice, interesting kind of USB shapes coming from Oxford and UK for also sequencing. So it's going there, and hopefully it's gonna, some technology is gonna break this even $100 genome barrier. Novus 6 series represent Illumina's most advanced technology. It offers flexibility in terms of read length. There's one of the things, so if you are going to look at these different technologies, one of the things that you want to look at is read length. Okay. Another treasure hunt question, why do we care about read length? Okay. Like, in other words, if we have longer reads that we can read through the technology and or shorter reads, anything that relates to what quality and how. Okay. Flow cell options, there are many flow cell options in terms also of the developed technologies. These are the glass lanes that helps, also to capture and do the imaging from different technologies that we just looked into. Okay. Also here's this is in terms of price per gigabase of reads, increasing system price and output. Okay, so there is going to be increased system output. Okay, and help in building lots and lots of experiments. Okay, looking here at 167 to 6,000 gigabytes. So this is certainly huge throughput. We're looking at 2 times 150. I'll see this numbers later on. And this would refer to, we can do forward, reverse strand, okay? And this is a huge sequencing power, okay? That we are gaining through the recent developed technologies. This table summarizes some. So maximum reads per run, for one run, it can achieve 400 million using NextSeq, okay, or 20 billion. Imagine this using NovaSeq. This is really big. This is really huge. We're referring to 48 human genomes every 2 days. 48 human genomes every 2 days. It's huge, right? 300 base pairs of read length, okay? So we're looking at two numbers, the read length, okay, and the number of reads per run. Both indicate is going to directly relate to quality, okay? If we have, higher read length and higher numbers of reads, as we keep adding more reads, this reminds me of when we have done fragmentation. When we talked about in the previous module, this, should gone kind of ideas, or this newspaper example puzzle, where we get into many pieces. As long we have more and more and more and more repeats, it's going to make it easier for alignment for us, makes it higher quality, not only easier, but also high quality. So in terms of the sequencing that we see there, pair end, there are two types of sequencing, okay? There what's called pair end, which is, in terms of recent development of technology, it's more common nowadays to hear about pair end. So when we get the files for one sample, we get two files actually for each sample, one indicating forward and the other one is indicating the reverse, okay? In any way, this forward and reverse is gonna indicate the outputs files we get. But in terms of sequencing, we have paired end sequencing and single end sequencing that are used in next generation sequencing platforms, okay? And like those developed by Illumina. Now, each one, again, would have some advantages and a specific application. In single end, when you look at single end, only one end is gonna, only one end of each DNA is gonna be sequenced. Okay, which gonna give us what? A single read for each DNA fragment. That's what we get, a single read for each DNA fragment, as compared to two separate reads for each DNA fragment, a forward and reverse read that I've just referred to through the paired end technology. So in paired end, what you expect, you have a forward and the reverse. Remember the DNA directionality, we have forward and reverse. So this is what we're going to end up with pair end sequencing. Okay, from two ends there are going to be some read. One is the forward, and we'll have this in one file. Okay, one file, call it file R or F forward, and then we'll have this output in another separate file called file reverse. That's the paired end. Okay, and in between there's what's called the insert size. Okay, it is the distance between the two sequenced end, or the size of fragment. We call it insert size then. Okay, now eventually there's going to be an alignment step. So this alignment step would help to look into the unsequenced region. Okay, so because in paired end sequencing, both ends of DNA fragment, they are going to be considered are sequenced, and they will result in two reads. Okay, sorry. The middle portion that you see here, okay, in this case, pay attention, the middle portion which lies between the two sequenced ends, this is what we call the insert, is not directly sequenced, but its size and sequence can be inferred based on the alignment of the paired end reads to a reference sequence. So this is going to be inferred. Okay, we will see some examples very shortly. So as we looked even in the previous, similar to the previous animation, so we will have, as part of the library preparation, we could have different libraries together, okay? And each one would have its own barcode. So this is CATTGCG. So the file, output file at the end, should give us for what is the index. Okay, this is the index. Because index eventually is going to help us group some reads together. Okay, it's going to help us to group some reads together. Okay, and so to be able to align those, okay, for one library or those for another library. We don't want to make mixing. Okay, we don't want to make mixing. While we can mix through the experiment, we can still separate them. There we can mix them in the pooling, sequencing step, but then we can separate them, which is nice. And again, this means what? Why we're doing this? In fact, the question is why we don't do it. Okay, why we don't do it? If we can have a technology where we can mix up some things, speed up the process and scale up the process, and then also we guarantee that we're gonna end up, being able to separate these samples, that would be great. So there is this demultiplexing step, and after sequencing, we will have raw data which consists of reads, okay, sequence output to data file. So this is the output we get from the sequencing step: library preparation, pooling, sequencing. This is the output file we get. Something we'll have access to. We'll see a sample today. This looks a nice output. Maybe the sample would have a little bit more details. We're going to look into that. And then demultiplexing is the process of sorting these reads based on their unique barcodes. These are the barcodes. Okay, these are the barcodes that we had. You want to call them barcodes, you want to call them index, right? You can do so. As well as adapters. Okay, we refer to these as part of the sequencing by synthesis as terms adapters. Okay, and this demultiplexing again, once we are able to sort these, we can separate the data for each individual sample, which is key. Okay, so we'll end up with a set of sequence files. Each one is corresponding to the original sample. So while we get one file, we can actually separate this based on the demultiplexing and then do alignment next. So we'll have one for library 1, library 2, and those who are in the biotechnology side, they would know what is library 1 and library 2. They have prepared one experiment and then another experiment and so on and so forth. Okay, and then for the Illumina sequencing steps, as sequencing is done, the different cycles are repeated. We get an image, a digital image. And again, as a new nucleotide is added at each step, an image is taken. This is one cycle. It is the process that is repeated. We end up having the data, which is exported, indicating the different clusters and the read outputs. And again, we'll have these reads. What we need to do with the reads, once we are able to group them, categorize them, and find these are the library 1 cases, okay? We want to align them. Now, alignment is one of the very common and one of the well-defined computational problems in bioinformatics. We're not diving into this at this stage, but what we're looking at is how we can categorize those on top of each other. It's one of the fields in computer science, and specifically in under the topics of algorithms, a very interesting class of algorithms. How can we find the aggregate solution of all of this? Okay, and again, we have a reference genome that we are trying to map these reads into, right? So there are areas of overlap, which is, you know, like this is easy, right? It's all G's, so this seems G, so we know that this is the position. And we keep going. These are 2 T's here, so this is another 2 T's. But we have some differences in some areas where we're gonna need to ask some questions. Why do we have such differences? The sequencing technology there, as part of Illumina, initially they had 4-channel chemistry, where each nucleotide has a specific fluorescence, so 4 fluorescence. So 4 colors, 4-channel chemistry. Extra images, okay, or extra imaging channels, image 1, 2, 3, 4 channels. Now, the newly developed version is using 2-channel chemistry. This is going to reduce the imaging channels and help in making the process more efficient. How it's done? Well, we can give one fluorescence color for 2 nucleotides, another for other 2 nucleotides, and then based on some variations, we can explore which one is which. For example, this is green, this is red. Okay, this is a mix of green and red, and there's nothing, so it means guanine, and so on and so forth. These ideas or these variations along the different cycles of imaging can indicate to us what we are having. So it is increasing efficiency. So here we come into another concept here that is called hybrid capture or target enrichment. Hybrid capture or target enrichment. This is a method to selectively isolate specific sequences of interest from a complex DNA sample. In a nutshell, the idea is if you are interested in targeting specific parts of the genome, for example, what's called exome sequencing, okay, exome sequencing, where we are only targeting protein coding regions of the genome, okay, exons, that is what meant exome. Exons then are the areas of interest, or other types of pre-designed regions. So if we're interested in looking, then we'll have a pooled sample library, right? How we make sure we get these specific reads related to that specific area of interest. So here what's interesting is having biotin, which is a kind of protein, and streptavidin, another protein, and those are well known to bind very strongly to each other. So it's kind, you know, we're gonna attach send biotin with specific designed probes to target specific regions, and once it binds to it, we're going to send the streptavirine PEDs, and under certain magnetic resources, it's going to pull these reads for us. So out of millions or billions of reads, we're going to pull these reads that are related to specific area of interest. So here with some modification to the Illumina-based sequencing and to this high throughput sequencing of next generation sequencing, we can do this to even target specific areas of interest. One of the common issues that I've referred to earlier when it comes to next generation sequencing and Illumina sequencing is exactly the example when I said, you know, if I stand up and my kid stands behind me and we both send some light source, if you can see it or not, if I'm going to block my son, exactly the same thing. For a certain reason, there is an issue that could happen because some DNA strands within a cluster get out of sync during the sequencing process, and so we end up with this, okay, which means different lights. Think about my example and my son. This could block this, or this could affect this. This is going to generate some noise. It may happen, yes. It's not intended, and it is this situation where some DNA strands within cluster fall behind the sequencing cycle. So, for example, a nucleotide fails to incorporate into a strand. It could happen, like, okay, one nucleotide, you know, fails to get into the specific position there. And actually, there are two concepts. There's what's called phasing and pre-phasing. So in phasing, a nucleotide fails to incorporate into a strand. That strand becomes one base behind the others. We don't have C here, okay? So this is one strand behind. So it's having gene, it's green. We see it's bluish. And the pre-phasing is actually have an extra one here. And those ones, what we're looking at is to have the same level for all of them. And then we take capture, we take our image, we are hoping we capture the light differences. And here I would link this to this animation here. At one cycle, we get the photo, and then we know what are having. But with this, we're going to certainly have some sources of noise. However, there are ways to mitigate this, because this signal that comes from out of phase, whether it's phasing or pre-phasing, is going to introduce some noise, which, you know, is going to impact the outcome. Now, in Illumina sequencing, it uses sophisticated algorithms to correct for phasing and pre-phasing. In a nutshell, these algorithms estimate the rate of phasing and pre-phasing for each cycle and adjust the base calling accordingly in the following, using the following idea. We have the intensities of G's, C's, A's, and T's, okay? And based on understanding the different intensities, we can do different corrections. Because again, this is what we are not hoping to have these big differences, right, at the specific instances or cycles. So here we get an interesting raw sequence output, what we're looking at here. So this is a FASTQ file. This is R1. This is forward. This is R2. This is reverse. So this is the name of the file. So this is taking from head scratch into some file name. So this is named AIP02.R2, the reverse.R1, the FASTQ. So we have two FASTQ files. This is the once you try to view part of the file, usually we don't open these files on our machines. Okay, they are not regular text files. Each one could be roughly 2 gigabytes of information. And so if you just end up, you could hang up the word processor that you have, or maybe the machine. So we usually try to use the shell programming for that. So we view part of it, or we use some programs to try to extract the information from. So what do we have here? What is the first line here? That first line starts with an @ Okay, that first line starts with an @ for the forward and reverse. This character here indicates the sequence identifier. So what is going to come next? Help us to identify information. about the sequence. So this is line 1 is sequence identifier. Okay, we have sequence identifier here and sequence identifier here. Part of that is the sequence ID, which is coming here directly. So M0, 20, 58, 32, and so on and so forth. You get up this number here, and 58, 30, 62, and you keep going. Okay, we have all of this information, and then what do we get at the end here is these numbers are some of these numbers are explaining coordinates, okay, and like these numbers in the middle, okay, may indicate what referred to as coordinates or identifiers related to the specific location or cluster. Remember the clusters that we had there on the flow cell? These are having specific numbers and coordinates. They are also part of this. So this is in general, this line here is giving us access for sequence kind of meta information, sequence identification, sequence information. At the end here, we have the index. This is what we refer to it as the index. Remember when we had this to group the different libraries? So the index here is TAGCTT in all of them, okay? So we have some information if we're interested to query and so on and so forth. This is 2, giving the reverse side, okay? And here we have the different location, 40, 47, 54, 71. We have some different numbers here, and so on and so forth along all these positions as well. Okay, so some meta information. What is this here? The second line, always when I say the second line, it is the second line, okay? It's not random order. This is specific format for FASTA-Q file. The second line here is the row sequence. This is the actual nucleotide sequence. It could be DNA or RNA, depending if we see uracil here or if you don't see uracil. So this is the actual read, okay? And then we have a plus, which is a separator, a line separator. So this is a plus here. And line 4 is giving us very important information. It is the quality score. The quality score. This line contains quality score for each nucleotide in the row sequence. This is going to give us a quality score for each nucleotide. Okay, for each nucleotide. It is written, sorry, not this one. Okay, this is written in ASCII format to make it more readable or compact. But certain information that we can extract. So, for example, when we come to the RNA-seq lecture, we'll see how we can extract this information and visualize the quality information. This is very important information that we're going to look into. Okay, let us dive into this. So you can see here, okay, for this first line, we have some information. So you can review this through this table as an example. Unique instrument name, all are going to indicate the sequence identifier, and then yes, the index sequence at the end. Now, what is this formula we have here? ASCII code and the quality score. Okay, so you have ASCII information here that's going to, as a reference, you can decode that or unhash that, whatever, or translate that and get the quality score that we're looking at, which relates to error error probability. Okay. Error probability. Let me give you perhaps an example. Okay, let me actually share some example with you here with what's called Fred score. Okay. Bear with me. I'm gonna look something here to have the right numbers from my notes. Yes. Okay. So let's do this. So there's what's called Fred quality score. Okay, Fred quality score. The quality scores in FASTQ files, the one that we just were looking at, are often represented using these Fred scores, which is, as a score, it is logarithmically related to the base calling error probability. So we have one, as we said, we have a quality for each base. So this is usually Q equals minus 10 times log, okay, base 10 of the P, the error probability. So if the Fred score equals 30, for example, okay, how do we read that? It indicates Fred score of 30, it indicates an error of probability of 10 to the power minus 3, okay? Or in other words, this is, we can read it 0.1%, okay? Meaning, in other words, that it's 99.9% that the nucleotide, the specific nucleotide, the chance that the nucleotide call is correct. So imagine that specific position in that long read. We had here A, and then we have the score here. The Fred score is 30. This means it's 90.9% correct, and so on and so forth. Usually we use plots to show this whole information. And this is again indicates the relationship. As you just said, 30 is going to be 99.9% of accuracy. The different technologies are out there. This is MinION that we just explained. MinION can work with big, very long reads. Okay, yet error rates are high as compared to the Illumina. Now, as types of errors that are common in Illumina are majorly substitution. So there are substitution, insertion, deletions as kind of errors that may happen, sometimes random errors. For example, this slide here highlights some of the errors that one may face with the Illumina technology, okay? So there is this error here, okay? This error, for example, is just a random error. It's likely when it's close to the end of the sequence read. Okay, you see the read is happening this direction or this direction. So towards the end of the sequence read, there can happen some errors. Okay, due to higher chance of errors using the Illumina sequencing or technology, for example. There could be some sequence-specific errors, some kind of random also. We have some C's here, and then we end up having, you know, some C's. So this could also end up causing an issue while reading the specific sequence, substituting that. There could be some systematic error, like this third type of error here. Systematic error means it consistently occurs under some specific conditions. So there are some errors, for example, associated with DNA motifs, like having, if there's GGT, okay, GGT, then we expect that there can be common over, or GGC. These can usually are linked to phasing issues during sequencing. And we just looked into the phasing issues, right? So we may end up with some errors during sequencing or the machine sequencing. Now, if we start aligning all of these reads and look into this example, the reference genome is saying this is a T, but the alignment here is saying mostly is, if I'm going to use like the alignment output and based on the statistics here, this is the best attempt that we have through the algorithms applied. So these are commonly C's. So is this a substitution variant or a substitution error? Okay, is this a substitution variant or a substitution error? Substitution variant and substitution error both involve a change in the nucleotide sequence, but they differ in their origin and how they are addressed in genomic analysis. So think about it and share with me your answer. What first comes to your mind when I say substitution variant or substitution error? Okay. And write it down with your own words. Then again, substitution variance, we could relate it to species level, right? And something that could be under the evolutionary ideas, or maybe it's an error from the machine. It is one of the very difficult questions to answer in genomic research. We could have insertions and deletions, like in this case, a deletion. So there's a missing information here. And again, we're trying to get this information through alignment. We could have large deletions and so on and so forth. So hopefully with this part of the lecture, you were able to capture the next generation sequencing, Illumina sequencing in particular. Make sure that you are aware of these terms that are in front of you in this slide. We just looked into an interesting example of Illumina sequence file, looked into the information we have. Personally, as a bioinformatician, when I look to this file, this is the part that I'm most excited about. Finally, we have something that we can read, and hoping sometime soon we can start to putting our hands in this and then running something, right? Some sequencing, some alignment, or reading some gene expression, whatever. Okay, and thanks so much. We're going to continue the next section.